Good afternoon again to all of you. So, basically, I'm Suvanjan Bhattacharya from the Department of Mechanical Engineering, Pilani campus. And as I mentioned, that we are suffering with the proposal together with Prof. And along with me, more colleagues from the Indian side, and we have to. So, this is the third day, and I believe that the workshop is the grand success because from the first to the last lecture, whatever presented by the keynote speakers, if all of the lectures were really good and very engaging. So, I'm basically a thermal on the fluid side guy and working in the area of thermal heat transfer, right? So, it's a very new topic that we are working and which is very relevant to the fluid dynamics. So, I'm trying to present some simulation work and also we will talk about the thing. So, basically the presentation will be divided into two particular parts. First, we'll talk about the nanoparticles diffusion in the tumor micro environment, and then we'll talk about the microbiology of the circulating fluid. So that I've just worked with Professor Rajdeep, Professor Samantha. They have presented. They are my colleagues from same university. They're from the biological science department. I'm taking care of the mechanical part. This is the university, Pilani, which is one of the top university in India, and mechanical engineering national rank is 10. So this is our main building. This is the clock tower. This is very iconic. It's a very old university in 1969, and this is the new academic building. So Professor Samantha was presenting yesterday. Here is my office. Okay, and very unique is that we have a garden at the top of this particular building, and this is the part of Rajasthan, which is the western part of India, sharing the border with Pakistan, and it's a very hot weather, hot and dry. During summer, the temperatures would up to 50-52 degrees Celsius. In winter, the temperature is -1, 0, -1, but there is no snow, right? So we have a designed such a way. And another important thing is that this building, this tower, and just straight opposite, we have a Saraswati temple. Goddess Saraswati is for study. So it is just opposite, and there is no barrier in between. So our director and the vice chancellor sits here, and just straight opposite is a temple. So it is like we worship the God and directly is a connection with the God. Okay. So yeah, this is our library, and this is the mechanical engineering department. So this is the mechanical engineering department, and this is on, and this side is my lab. So I welcome everybody in my university, and it's a very beautiful university, and everybody will stay inside the campus. All the students, all the faculty members, their families, everything inside the campus. We have the supermarkets, everything. So all the facilities inside the campus. You don't need to go outside. So inside the campus is a city, that kind of thing. These are the collaborators that I work together. Rajdeep, Professor Rajdeep, Professor Sudeshna, Professor Samantha. Professor Sudeshna is the wife of Rajdeep. They work together in a group from the biological science department, and I work from the mechanical engineering department. These are the two very bright students. So it's Naman and Surjo. So Naman is just graduated. He's undergraduate student. He graduated and worked with me for the last 3 years, and now he joined the masters in Georgia Tech in West. And Surjo is currently 3rd year, and he's very much interested to join the masters if you have a funding. Both of the professors. He's a very bright student. He's worked with me for the last 2 years, and is looking for the masters. He's the last year. He's been graduated and looking for a funding. So if you have anything, I will recommend him to. He's a very good guy. So as the workshop is concerned about the tumor cells, or basically the cancer cells. So if you look into this particular graph, you will see that starting from 2022, and we are predicting up to 2050. Okay. And if you look into this one, this is estimated number of cancer deaths. So you see, each and every year, it is increasing. It is increasing. So if you look into this particular figure, in 2022 is this one, 23, 25, And then, as this particular world is concerned, it's going with lots of junk foods and all these things, alcohols, and so people are going crazy with all these foods and all these things, right? And it is predicted that it will go this particular figure in 2050, which is very alarming, right? So it is very important for the as an engineer and as well as the biologist, people to come together to find out a solution. So there are several types of cancers, and if you look into that one here, you will see that the figures, what we have presented in the previous slide, are like this. These are the number of percentage of the cancers the people are dying, right? So if you look into this one, lungs, trachea, and the bronchi is the major concern, which is the colon and rectum, then stomach, then breast, and also on. So my work will be a little bit regarding the breast cancer. We will talk about some tumor cells in the breast cancer. So over 20 million people are diagnosed with the cancer annually. And this number is projected to reach 33 million by 2050. 2050, it will go to the 33 million, which is very, very alarming, particularly this figure. This one? Thank you. So, cancer remains a leading cause to death in the worldwide, posing a major health challenges. Population growth and aging contribute to rise the cancer. And a country like India, country like China, the population is huge, right? And, in India and China, the senior citizens are also a huge number. Right? We have to concern about this as well. So nearly 1 in every 10 people expected to face the cancer risks in the coming decades. Okay. So there is a strong need for innovation and sustainable treatment strategies so that this figure, we can restrict, we can minimize this particular figure. Then rapid multiplications of the tumor cells leads to the tissue invasion and the metastasis. Everybody know we have to speaking these things for the long time from the beginning of this workshop. Then bone marrow metastasis is actually the process to the complication treatment. So if you look into this particular figure, this is the first stage. Cancer originating somewhere in the body. Then cancer travels to the blood, where the CTCs comes into picture. And then the cancer goes in the bone. And then the typical effect of the people are breast cancer, prostate cancer, lung cancer, and so on. Right? So there is a need for selective and minimal these alternatives to stop this particular cancer, which is spreading around the human body. So emergence of the magnetic hypothermia, fluid hypothermia, is very, very important and selective to heat the tumor cells, which can 40 to 45 degrees Celsius, which can kill the cancer cells, which is very important for us to understand. The temperature can be reached up to 40 to 45 degrees Celsius. That temperature can kill the cancer cell. So healthy tissue remains largely unaffected. So initially, concept is 1950s back that nanotechnology comes into picture. But now in the model world, we have to use this nanotechnology in a very wise manner so that we can kill and treat the cancer in an effective way. So in this particular study, what I have done so far, in this study, we have used the magnetic nanoparticles, which is Fe3O4, which is biocompatible and magnetically responsive, exhibits strong heating efficiency under alternating magnetic field, widely studied for localized hypothermia and the targeted drug delivery. In vivo studies that on local heatings are still limited because we have to do it very systematically and the planned manner. It's not the arbitrary way. So we will show that how we can increase the heat of the cancer cell to kill the cancer cell. Then real-time monitoring of temperature, nanoparticle motion, and diffusion is very, very important and sometimes it's very difficult and challenging in the literature that we have found. So we have tried to solve this particular point as well, and need for a numerical and simulation based studies to bridge this gap. So in experimentally, and there are some challenges. So first we can do the simulation to try to understand the physics, how the fluid dynamics will there, and then that can be converted into experiments, and we can be successful in the experimental studies as well. So to explore, so you can say the motivation and novelty to explore how magnetic field and the body vibration jointly enhance the nanoparticle heating. So this is very important in this particular work. Like people are studying the simulations till now, just only the nanoparticle heating. And there is no the simulation of body vibration. Right? So in our body, there is some vibration, always the vibration is going on. Because blood are consistent flow. Right? So that vibration, body vibration, we are incorporating the body vibration along with the magnetic field to see how much temperature can be generated and can damage the plant. Inspired by the previous work, Suleiman or I told in 2022 work similar way, but not incorporated the vibration. We have taken the models and everything from that previous study and to establish the more realistic and the proper mechanical models here. Introduce 5 Hz body vibration in an additional factors to the magnetic field in our study. Propose magnetic hyperthermia with a mechanical microscopic effect. We are looking into this particular study. So these are the properties that we have taken from the literature. That's the magnetic nanoparticles Fe3O4. And we have used this particular magnetic particles in our study over here. So this is the model that we have used in our study. So if you look at this particular model, we have tried to analyze 3 different sizes of tumor cells. So the first one, the small one, so that is a micrometastasis state in a tumor cell. Then the little bit of bigger than the smaller one, we have a priming models are tumor cells over here, and then we have a mature tumor cells, a bigger one. So 3 different types of cells we have used in our study, and the nanofluid inlet will from the artery and this going down to the vein, right? And then we call this particular things as a tumor cell microenvironment. Yeah. So realistic inlet and outlet correspond to the artery and the vein respectively. The gap in the geometry corresponds to the tumor cell, and intentionally use cell of the various sites to recreate the realistic tumor microenvironment by using a combination of the microaroma and the mature large tumor. So we will try to see that in a small tumor cell, what is happening with the kidney, with a little bit of larger stage, what is happening with the kidney, and for the a little bit of bigger stage, how much the cell can be damaged. So that is the reason intentionally we prefer 3 different types of cell. So when we do the simulation, this meshing is very important, as you know. So we have done this particular meshing by using ANSYS only. And we have also used different size of a meshing, right? So if you see, look in the fineness, the normal, close, closer, extra close, extremely good. So we have tested our work with all the different types of meshing, and we found that for the normal, fine, and extra fine, with comparison to that, there is only 10%. There is not that much of difference. So, and for the extra fine to fine, the results are just around 1%. So we use this fine here for our study to reduce the competition. And these are the equations that we always need to solve. And it's a laminar flow. So we are using the laminar model. So, and these are the equations that everybody be able to understand. And when we added the vibration, this particular vibration term has been added. And this is the heat for the magnetic nanoparticles. Then we have used 10 different locations. So A, B, C, D, E, F, G, H, I, J, K. So we have 10 different locations of the tumor cells, and we try to find it out that how much you think will be there in that localized area. Okay. So the objective of this numerical study to create the solid foundation for the study for the FH. In light of that, then the present study analyzes the pressure drop, localized temperature gradient, and the velocity contour, and the local temperature gradient measured in 10 different tumor cells location, and monitor systematically till the microenvironment attains the steady state. So these are the boundary conditions we have used in our study, and we have used the porosity of this one from the previous literature, and also we have used special distillation. Okay. So after doing this analysis, if you look into this particular figure carefully, the first one is the pressure and the second one is the temperature. So near this particular film, the pressure is a little high, and when it's flowing through this particular tumor microenvironment of the tumor cell, the pressure are eventually distributed. Okay, and when this pressure is eventually distributed over here, so a little bit high and then small and decreases slowly. So there will be a velocity. Is also, when it comes to the velocity, you will see the temperature when the pressure is there, and then the temperature is also increasing. Here and here. So here we have a high temperature, the fluid is inlet from here, and then it will distribute here. So maximum portion of the fluid is flowing through this one, and the little bit of low pressure is coming down here. Right? So, and then due to the Neal effect and the Brownian motion, the temperature is increasing over here at the surrounding of the tumor. Now, if you look into this one, next one, then we'll see how the fluid flow, nanoparticles and fluid flow around the tumor cells. So you see that there is a small swelling effect is coming into picture, and the sitting time inside this particular tumor cells will be more, so that it will come in contact with the microcell, different sets of tumor cells, and it can generate a lot of heat over there. And due to, as I mentioned in the previously, 40 to 45 degrees Celsius, that particular temperature can destroy this particular tumor cells. Now, if you look into this temperature with time, this is in minutes, and this is a temperature in Kelvin. So, as I mentioned, that at 10 different locations, we have studied this particular microenvironment of the different tumors. So, 1 to 10, you see the temperatures are increasing. This one is only without vibration. We have not incorporated the vibration till now. The temperature is around 315. Right? And for all the part portion. And then if you come down here, which is the zone 1, this is here, our temperature is lower as compared to 10 over here, 9 and 10. Because once the flow here, and due to the recirculation, the cell which is a little bit further from in, having much higher effect, heating effect, and the temperature is higher as compared to inlet of heat. Right? So then is good. Also, thermometer zone 1 to 0.9. No, everywhere the temperature increases. What I mean is that at zone A, whatever the temperature is increased, there will be a significant enhancement of the temperature at the downstream of this. Because if you go back to the previous slide, you will see that there was a recirculation and the fluid is having a small vorticity generation over here. And due to that, the fluid will be sitting more inside this microenvironment. And that can interact with this particular cells more and can enhance the temperature. Yeah. Now, this is without vibration incorporation. This is with the incorporate of vibration, right? Now, if we add a vibration of 5 Hz in the system, you will see that there is a significant enhancement. If you remember the previous graph, the temperature over here is 315 degrees Celsius.